Hi, and welcome to the Anti-Waffle podcast with me, Bellshaw-Naylor, and Marva Gregorio de Souza.
Yay!
And together we'll try to talk about research about any gobbledygook. Each episode we'll be talking to a researcher who will discuss the work they're doing, looking at why this research is important and asking how the hell did they get into this field. We hope that it will be informative and interesting and bring a bit of the academic world into the real world. This week we have Professor John Christodoulou, Professor of Biological NMR and based at the University College London. Hi John, thank you very much for being here and thank you for talking about your research. Would you like to start and tell us a bit about your research?
Yeah, of course. So I'm a structural biologist is the overall term, which means we look at the ways in which we look at the cell and we look at the workhorses of the cell and try to understand how these workhorses of the cell are actually made. The ways we try to do is we try to understand things in molecular detail. So we really try to understand things at the very, very most basic level absolutely possible. What that actually means is that, in the same way that we can see things with our eyes, we kind of take pictures of things that the eyes and microscopes can't see, essentially. We're looking at biological molecules that are 10 million times smaller than a pinhead. So we're taking atomic photographs of those. And I use a technique called NMR spectroscopy to be able to do that, which is a spectroscopic technique, which essentially means that we excite the atoms and they absorb radio frequency, and as they emit their radio frequency, the radio frequency that they emit depends on their chemical environment. So they give up lots of clues about their chemical environment in using NMR spectroscopy. So what we try to do is we try to understand the ways in which biological molecules are made. So within the cell, so the cell is the basic unit of activity within all organisms, and in order to do that the cell needs to be able to make its products. Right. And in the same way that society has machines, engines, plows to be able to carry out its work, the cell has certain machines that are molecular machines that do its work. One molecular machine is it's called the ribosome, and the ribosome is something that's present in all kingdoms of life. It's composed of proteins and nucleic acids, and its job is to take the DNA code that we inherit and turn that DNA code into these workhorse proteins. Right. So it reads the genetic code and then it spits out proteins at the other side. And we need those proteins, we need hundreds of thousands of those proteins. There's millions of them being made in your bodies at this very moment in time because they need to do all the work that the cell. For me to be able to speak, we need proteins. For me to be able to see, for my blood to circulate, for blood to exist, we need proteins, and they need to be continuously made and turned over. Our skin, etc., etc. So we need lots of proteins being made, and this molecular machine makes these proteins through reading the genetic code, and it has to do it accurately every single time. Otherwise, we risk a set of diseases that we can discuss. So in reading the DNA, it produces these chains of proteins that are chains of linear amino acids. So these are the basic protein building blocks. Right. So nucleic acids are the building blocks of DNA, but amino acids are the building blocks of proteins. And amino acids are strung together one after the other to create these proteins. And there are different types of amino acids that come together. There are 20 types of amino acids, and they come together in various orientations to create these proteins. And these proteins can be, have 100 of these amino acids, or it can have 1,000 of these amino acids within their sequence. Now, this ribosome machine produces these amino acids, produces these proteins as a linear sequence, which means that it just produces it as a string, essentially. Right. The string has to somehow fold into its correct three-dimensional shape. Now you can liken that to the art of origami, for instance, right, where you have a flat sheet of paper and there are specific instructions to fold it into a boat or a hat or whatever it is you're making with origami. Well, the cell has instructions to take the linear string that the ribosome produces and turn it into the particular protein shape that's needed, and that's all within the amino acid sequence. The amino acid sequence tells it how to do that. So
it's like a IKEA flat pack, and it comes with the instructions with it.
Yeah, absolutely. That's exactly what it does, and it's been developed by evolution over hundreds of millions of years to go from very, very simple proteins to the kind of extensive range of proteins that we need for the higher thought that we, at least in principle, have at this moment.
How does it know which ones it needs to produce when?
This is
or is that what it's studying?
That's a really fabulous, a really fantastic question, right? It's a heavily controlled process. It's called the protein homeostasis of the cell, which means that there's a whole balanced network of recognition and checks and balances to ensure that the right things are made at the right time. So if we have a particular injury or a stress, then there'll be messages sent to the ribosome to start making some more protein sharpish because it needs it in a particular part of the body, and that's sent to the part of the body.
Right.
So, and it needs to be the thing that we really look at is we try to understand how this linear string that gets produced folds up. So we're trying to understand what the rules are for the origami within the cell.
Right.
So you're trying to find the instructions for all these different shapes.
Yeah, essentially. So, and this happens because the process of synthesis, the ribosome is making its proteins. This is a relatively slow process, so that the process of origami actually occurs while the protein is still attached to this big machine.
Right.
Now, so this is a relatively new finding over the past decade or so, and it's technically called co-translational protein folding. And every protein needs to take up its unique three-dimensional shape. It needs to take up a particular topology or shape in order to perform its function. If it doesn't have that function, it won't be able to undertake that function. It won't be part of your blood. It won't go to your eyes. It won't go to the right place. It won't function in the right place. And this is the best examples of the problems associated with this are there are a set of diseases that result from essentially the origami process within the cell going wrong, and they're called protein misfolding disorders. So common Alzheimer's disease, Parkinson's disease, type 2 diabetes, so very, very common devastating killers are associated with the process by which the molecular origami in the cell doesn't occur appropriately. And what happens when it doesn't occur appropriately is that you have these long strings of proteins that haven't sort of folded up into their correct shape.
Yeah.
And as a result of that, they start to stick together. One starts to stick to the other because the body can't cope with the idea of having these long strings. It doesn't like it. There are too many things exposed that are potentially toxic. So lots of them come together and form these plaques and tangles, and they're amyloid fibrils. And these fibrils, which are now big clumps of protein, so rather than these beautifully elegant little shapes that the body will normally make for proteins, these clumps are also rather beautiful, but they're completely functionless and toxic to the cell.
Right.
And they deposit within, for example, the brain. In the case of Alzheimer's and Parkinson's disease, you've probably heard of the idea of plaques that occur. In other amyloidoses, you get complete. The amyloidoses are the diseases associated with these states, with many of the protein misfolding disorders, and you get massive deposits of proteins that are aggregated, that are clumped together in the body, and this is normally the pathological kind of hallmarks or fingerprints of disease. And so by trying to understand the process by which the molecular origami actually occurs within the cell in molecular detail, and why the whole balance network that you asked about goes wrong. There are processes in the body, like there are chaperones, they're called chaperones, they're molecular chaperones that try to help proteins to do this, to find their right shape.
Right.
As we get older, or if we're under disease or stress conditions, these chaperones don't work quite as well. And that's the reason why, as we get older, we're more likely to get a set of molecular disorders. That's why Alzheimer's is becoming so prevalent because we have an aging population,
Right.
but the body can't cope with the ability to make proteins in the right way.
Wow, that's fascinating. So, how is your study coming along? What are you? Have you discovered anything?
Have you found any of the instructions for your blood banks?
I think we very much have actually. I mean, when I started this research probably about sort of 8 or 9 years ago, I remain absolutely astonished by the fact that how little is absolutely known about the process. You would think that this is one of the things that I think is really amazing about science in general, is how little is known about these key processes. The fact that each of us need 1 million proteins to be continuously being made, and yet we don't understand the process in any significant detail absolutely astonishes me. And so the extent to which it's been studied has been very, very poor. And I think that if you actually touch the surface of most of the research that occurs, you find that actually very little is known about most things, and that's actually quite shocking. And I think
Do you think that might be a technological reason for that? Like, because the technology is not there to study something. Like, before microscopes were made, we didn't know anything about microorganisms until there was a microscope to look at them. So
absolutely, yes. I mean, I think that the advances in technology, the NMR spectrometers that we use have improved significantly. The microscopes have gone from light microscopes to very, very powerful electron microscopes that are continuously undergoing small revolutions. There was the Nobel Prize this year was in electron microscopy, which has taken the technique from being able to look at molecules as blobs to being able to look at them in intimate detail where you can understand atomic bonds. So the technology is significantly improving. Alongside this, you have computational power, because it's all about being able to handle the large amounts of information and process it very rapidly in the process. So the increase, there's this Moore's law, which essentially says that the speed of computers doubles every 2 years.
Right.
So that has gone much faster than that for several years. I mean, it's slowing down a little bit now, but that has meant that we can really analyze the data from microscopes very, very rapidly and begin to make actually rather rapid progress. So there's no reason why, for example, if we analyze the sample this morning, we could have a really good picture of the molecule we were looking at by this evening or tomorrow. So that's one thing. The other
I'm sorry, but that doesn't really impress me, because I'm used to movies where they'll just say, "Oh yes, I can." Well, no, in the movies they'll do that straight away, won't they? They'll say, "Oh, give me this. Can you test this sample?" And then they'll test it. It's like, "No, I can see what's in it," and I'm just used to
they don't get sent away for 2 days to wait for your results.
Put them try and make. I mean, it's a good point actually. But by comparison, these processes would normally take 6 months or up to 3 years, and the images you would get would not be particularly impressive. So the fact that you can do it very rapidly and actually begin to see details that you'd never be able to see before. I mean, astonishing differences in detail that have been able to be seen now through this technique.
Oh, that's brilliant.
I think on top of that, what's really happened is our understanding of, so that's the technology, that's the sort of big machinery and the computational part, but what really has gone forward is our ability to manipulate cells. So this is using molecular biology, essentially, we can really mutate and change and delete genes and look at the effects of these deletions.
Right.
There's a technique that we most people know about, it's this CRISPR-Cas9 technique that means that you can edit entire genomes. So, for example, with our molecular machine, the ribosome, what we tend to do is take the machine, look at its structure that we ourselves have got from electron microscopy and say, well, why don't we take this particular part of that ribosome away and see whether or not the origami process has changed. Right? See whether to see whether that part of the ribosome is responsible for helping with the instructions of that. And we're actually finding in many cases they are. Many of the, so we're actually beginning to tease out regions of this molecular machine,
Yeah.
which has over 7,500 atoms. So it's a rather large species, in molecular terms. We've been able to tease out the particular regions that are responsible for helping the origami process to occur in various forms. So, for example, the protein is made one amino acid at a time. It threads through this tunnel. It interacts. It starts to interact with various proteins within the ribosome, and some of them, it seems, would help it with its origami, and others are just there to sort of provide some type of support for the process. So by teasing these things are entirely unknown, so we're teasing them out kind of one little bit at a time.
What happens with
What we're very lucky to is that we're this has been the Wellcome Trust. This needs a huge amount of funding. We're very lucky to have a major Wellcome Trust grant in the form of investigator awards. So we really kind of hugely dependent on funders understanding what is important, but a lot more money is needed.
It's exciting.
It is, isn't it? It's like you said, there's actually a whole world of stuff still out there to be found, and it's just fascinating. You say it's just, you're amazed at how much hasn't yet been discovered or explored. That's exciting.
Beauty of structure, and I think that my main expertise is in the NMR spectroscopy, which is a whole technical thing by itself, and it's the means by which, in order to be able to observe these workhorses, especially when this process by which these workhorses are made is a dynamic process, so it's going on continuously, and so these things are wobbling around in space and doing all sorts of things. Now, it's very difficult if you've got a microscope to be able to take a picture of this static picture, because what you'll see is an average image. So if the workhorse is going one way and then the other, you won't actually see both of those states. You'll see some average state. So so techniques such as NMR spectroscopy allow you to understand not just the picture, but also the motions that occur.
So it's more like a video than
much more like that. So you see a range of structures that you can put together in a video of the process, and that's what we want to do. We're trying to provide a video essentially of the process of protein translation, which is the process by which these amino acids are put together by the ribosome to create these proteins. We want to watch the process by which each amino acid is made. We want to look at the linear string, and then we want to watch the process by which, once it's ready, it begins to sort of compact into its three-dimensional shape. And also how it doesn't do that in the case of disease states. How it can go.
What triggers it
to occur.
And by taking the bits out, you're seeing, well, is the trigger from inside or is the trigger external?
Absolutely, and I think that the technique NMR spectroscopy is an incredible technique where, in order to, as I said, what you do is we have these great big magnets that are probably half the size of this room, and we have to create the strongest magnetic fields known, and in order to do that we have to be at almost absolute zero, so we're at minus 273 degrees centigrade. We put the sample into into the center of this superconducting magnet, and then we hit it with radio frequency. And in hitting it with radio frequency, what we do is we do something that we call exciting the nuclei, the atoms. We excite the atoms, and then so we excite all the atoms within the molecule, and then we watch we excite them, and then we watch the process by which they go back to equilibrium. They go back to.
Oh, right, okay.
And within all of that process of the dissipation of energy, you can develop the structure. But I guess the reason to mention this is that you have to have this enormous technology to be able to even do things. So the development of these magnets, they're the order of £3 or £4 million a piece. You need a massive infrastructure. You need technical staff. You need continuous supplies of helium, liquid helium, and liquid nitrogen. So to be able to do this, there has to be a complete underpinning of the technology aspects that you mentioned earlier. And that's almost aside from the biology we were talking about. That's simply a physics kind of based thing in order to be able to use to put our samples into.
Right, wow. We've come a long way from just poking things with a stick. Let's see what you do.
But that's a great analogy. Actually, it's kind of what we're doing with fancier and fancier equipment.
Bigger sticks and bigger. Or to say bigger things, but littler things and bigger sticks.
Much bigger, slightly more expensive sticks. And,
is it just you, or have you got a team of you looking into this?
Yeah, there's a team of us. So I've got a lab over in the Darwin building on Gower Street. So, we've got these really lovely labs that have been actually purpose built for us, and I think there are about sort of 13 or 14 of us, maybe. I lose track of people, but there's probably about 16 or 17 of us by now doing this. And that's, as I say, the funding that we got from the Wellcome Trust, which is has been a 10-year grant that has enabled us to build this kind of infrastructure.
Right.
So we're very, very privileged to be able to do this. We're able to sort of select some really incredible individuals to be able to do this, and you need a diverse range of people. There's people that are experts in the NMR spectrometer that I talked about, so in the physics part of it, and in understanding how the machines themselves work, the big machines. And then you have to have molecular biologists who really understand how to manipulate DNA to make manipulations, and you've got I've got a computational biologist in the lab whose job is literally to sort of churn out structures using computers that are all over the place. We use high performance computing that is in either in Nottingham, in the United States, in Poland. So we've got calculations going on these very, very high performance computers going on as we speak, just because of the size of the ribosome and the process that we're looking at, and with every we really need these high power computers, because it just needs millions and millions of CPU hours to be able to understand the emotional processes that are going on.
Wow.
So these, and then you have students and all sorts of people coming in and out that kind of add to this process and come in with their own ideas. What's lovely about this is that we have this really broad range of, because we've managed to develop a kind of an area where we're recognised internationally for this. And so we have a broad range of collaborators from all over the place. I organised a conference recently where we had several of my collaborators in from Sweden, States, you might imagine, France, Netherlands, Italy, Greece, and just about everywhere. It was a 100-person meeting that became very intimate, and we're continuously sharing information, and we're competing, but nevertheless still sharing lots of information, and sharing DNA, sharing data, sharing all sorts of things. And that's a really nice part of it, a very, very international process, which kind of means that someone like me is very frequently travelling and hardly ever around, but it's very enjoyable.
Excellent.
And you said it's a 10-year funded project.
Yeah,
Where are you in that?
I've just started the second bout, so it goes until 2020, the end of 2022. So there's a few more years left to do this. We're very lucky to, it's highly competitive funding. We really fund about 30 people or so per year in the whole country to be able to, maybe up to 50 or 60 to do these kinds of projects.
Wow.
So we're very lucky, and they really select from a wide portfolio of research. And you can imagine that the work I do is one facet of the many areas of scientific research. So very privileged in that regard.
How did you get into this? Did you always want to work in this area, or did you have a different dream? How's your life got you to here?
Yeah, that's a good question. I came from I've always loved chemistry. I came from an area in London where science wasn't particularly kind of encouraged in schools, and I think this is something that I feel actually quite passionate about, and I'm kind of using an opportunity to mention it. I think that I came from an area in Tottenham, and it was just very deprived, and the result of that is that science in schools was just absolutely dreadful, and it meant that very, very few of us actually developed any scientific skills of any, or any skills generally of any consequence.
So maths and English, that seems to be the big focus now on schools. Oh, forget everything else, just get your maths and English, and it's just like, well what about everything else?
No, I think you're right. But even then, I mean, in this school I'm talking about, I think that even those skills were somewhat
dodgy. We were talking about how you were doing it, what you're with the research, and that it is international, and that even though you were working on it in competition, but then you're still collaborating. Yeah. But if we haven't put the investment in scientists from day one, from primary school, then we're not going to have the best scientists. And if we haven't got the best scientists, then we're not going to be at the cutting edge with research, are we? And so then people will be having conversations without England because we don't know what we're doing. And it's like, it starts at primary school.
I completely agree. I think that this is also, I mean, something where the investment in science in schools needs to change rather, increase rather significantly. I mean, the investment in schools generally needs to increase, and I think it has to be enthusiastic teaching. I think this is an important place, right? You get inspired by teachers at a particular age. There's probably I'd like to be able to pinpoint them myself, but there are several teachers that I think you were reasonably inspirational, and there was a particular point in time where I probably read something that took me forward. So I was actually naturally obsessed with things. So despite being in this rather awful school, I would spend most of my time actually not going to school and staying at home and reading my science books and getting in trouble for it. So it kind of all worked out somewhat. Actually, that's actually true. It was true for almost a whole term. I stayed at home reading my science books.
Wow.
So I think I was obsessed with chemistry and the process.
Don't do that, kids. I know it works for John, but it may not work for you.
So you were interested in this at school. So where did you go from you're taking your interest from chemistry?
So I loved chemistry, and I then went to university. I went to Imperial College here down the road from here to read chemistry, which I thought was absolutely fantastic, and I really enjoyed it. It was what I wanted to do, I think, since I was 11. I mean, I was semi-obsessed with chemistry books. I think what really helped me was in the third year of my degree, I spent a nice period in the Netherlands, in Leiden, and I made the switch from ordinary chemistry, if you like, just the understanding of bonds, to bringing in the biological aspect of it, so biochemistry, if you like. And that was the trigger for me to know that this is what I wanted to do with my future, that I really wanted to understand these processes. So really begin to understand the processes of chemistry that occur within the cell. And that's kind of what we're doing now. We're taking this molecular machine, the ribosome, we're understanding how the genetic code is read on it, but at atomic level, and how essentially this big ribosome, all it does is it catalyzes a chemical reaction between, there are two amino acids that are brought in by the ribosome, and they're in the same way that you could do in a lab, but very inefficiently. The ribosome brings them together and makes a bond between them, the peptide bond. And that's all the ribosome does, is it synthesizes, it catalyzes the process of making a peptide bond. Right. You could do this in a test tube, but you wouldn't be able to get anywhere near the efficiency that the ribosome does it in. But so essentially the ribosome is a big catalyst.
Right. Wow, so it's like a matchmaker.
No, it really is. It really is. And then all it does is it makes this chemical bond, and then it says, okay, I've had enough of you, move on a bit, and then it adds one to this and adds one to the end that's left, and then it continuously moves it on and continuously add on, while at the same time the instruction for that is coming from the genetic code that's just above it. Right. So it's reading the DNA on the one hand, and then in a kind of perpendicular to that is beginning to piece together these amino acids to make these chemical bonds. So I think that the process by which I then moved from using chemistry in the context of biology to be able to understand atomic processes that occur in the cell is what essentially triggered the process to get me to where I am now. And I've done various bits and pieces. I went and did a PhD. I was really lucky after that I was able to go to the Scripps Institute in California, which was in La Jolla in California. Again, the Wellcome Trust, the same body that fund me at the moment, funded me for 4 years to be able to do that. So I went to the States, and while I wasn't hanging out on the sort of, the beaches of California, I was actually taking this a step further.
Right.
Within this, I mean, I think that I need to mention the importance of metals. Metals are really, really important in the body: calcium, magnesium, cadmium, zinc. And so those, my chemistry background, in particular my strong interest in so-called inorganic chemistry, the chemistry of metals, meant that I started trying to understand how metals fed into the process of biology within the cell, and how we critically need selenium and many other body metals. I mean, we were working for several years. I was working on blood proteins, and I'm trying to understand how drugs that are based on gold and platinum bound to these proteins, and how they were influencing arthritis and cancer processes and things of that sort. So kind of lots of fusion of little things that brought me to where I am now. I think the big step, actually, that I think really cemented my idea that this is exactly what I wanted to do, was when I met somebody in Cambridge called Christodoulou. He's a professor in Cambridge. He's master of St John's College in Cambridge, and he was actually the first person I met that was utterly inspirational, who took me on and introduced me to the world of essentially of origami. Right? That was the first time I actually thought about the process of molecular origami and the processes that are associated with that. And I think that working with him, which I still do, actually, I was supposed to actually meet him right now, and I tell him I couldn't make it.
I like the fact you bumped him.
Sorry.
Actually, if his name is mentioned on this podcast, I can maybe send it to him so he doesn't think I'm just making random excuses to do this. So we still work together very closely as a close colleague, and he's one of these great minds that it's really nice to be able to go and spend some time with in one of these fancy oak-panelled Cambridge colleges. Very, very nice.
Brilliant.
Right, before we go on to another question, a thought popped into my head. With what we were discussing about the primary schools and the funding into the science thing, and the funding that you're getting for your projects and that other scientists are getting as well. How about if there was a system where you kind of had to pay a tax? So like your fund, when in receiving any kind of fund, that part of your tax of receiving that fund was going into the primary schools and actually either teaching, because the passionate people are the ones who are doing it, not necessarily the ones who are teaching.
So maybe not tax, but maybe as part of it a sort of public engagement bit.
Yeah,
do you mean?
yeah.
So maybe like an involvement engagement with primary schools or something.
Yeah, not a literal tax, just like...
Yeah, okay. I was trying to think of taking your money off.
Yeah, in return for taking this, any funds that you receive, I will donate some of my time, or we'll donate some of the money, or however you wanted to do it, into the primary school education for science.
I think that's a superb idea, and it's something that the funders, in particular the World Trust, have an entire public engagement set up. There's a whole department of that on that. It's a very important part of grant applications, which is increasingly taken less and less cynically by scientists initially. Maybe sort of 8 or 9 years ago, you'd fill it in without really having, really fulfilling every part of it. And now, we actually take it very, very seriously. And for example, I went on a public engagement masterclass on the Wellcome Trust up in Edinburgh to teach me just how to do this. And I then have gone to my son's school and talked to his form and a couple of the forms above them about what I do. And to be honest, I came in the same way. I was here somewhat unprepared, and the kind of questions you got from 7 and 8 year olds were quite astonishing, because I thought I showed them some pictures of some disease processes that were going on without being too gory, and tried to say to them.
They probably would have loved it being gory.
And the questions you got from them were absolutely fascinating. So we do spend actually a lot of time doing it, and I think increasingly more scientists are actually doing just that. So they're paying this tax, if you like. I mean, I think in terms of a financial tax, the funds that you get from these funders, although my fund, to put a number to it, is just over £2 million in 5 years. It just about
Colours.
things are expensive,
Yeah.
people are expensive.
Big sticks
Moreover,
are expensive.
Big stick
Yeah.
expensive.
big kit is expensive. So I think it should be the government that actually begins to fund science a hell of a lot more. I mean,
Right.
but the problem is that it is the question of priority.
Well, I think the public engagement idea is just great. The whole, because actually you're
Just get them
super
excited.
enthusiastic about your subject, and where else are you going to find super enthusiastic people but children who are just fascinated, and they will have the same enthusiasm. It's lovely, isn't it?
Just to be exposed to it. So,
Yeah.
oh, there's something else I can be
I
enthusiastic about.
I think there are many people, including in my lab, who are very talented at this process. Right? I think that my struggle with going into schools is that I tend to sort of start using jargon that is entirely inappropriate. But a lot of people have got this as a very natural talent, and I think that needs to be harnessed a hell of a lot more. Because I think if we're getting enthused, in the same way that we talk about very general things. Right? We talk about television, we can talk about the media, we can talk about all sorts of campaigns with Trump and other issues of society, feminist arguments. These are all fabulous and absolutely need to be discussed, of course.
Yeah.
But who talks about science? I would like to bring science into the same everyday framework.
Right.
If people can talk about science, if we could talk about my ribosomes in the same way we might talk about a Guardian article, then that would be great. And there's no reason why that shouldn't be brought into the general consciousness.
Yeah. No, absolutely. That's what we're trying to do.
This is what we're trying to do in this podcast, is to get it out there to people and to understand what's going on. So, and I think you've answered this lots of times, but my final question is, why is this important to the public? Why is your research really important to us, to Martha and me?
Well, I think that there's an interesting question that can be answered in several ways. I think that actually there's processes going on in our bodies that we don't know anything about, and isn't it part of the human spirit, if you like, to find things out about about the things around us, but in particular what's going on inside us fairly continuously. So I think just my personal main reason for doing it is to understand fundamental problems, right? So basic problems of the world, just really problems meaning questions of how things happen. How do we see? How do we open our eyes? What are the processes by which you're feigning interest in what I'm saying right now? Right? What are these things that are going on? How do these manifest? What's the molecular detail that allows this to happen? Because they have to be molecular processes. They can't be anything else.
Right.
Your body is telling you to do one thing. Your brain is sending a trigger of a hormone to one place to the next. It's somehow registering a memory, hopefully a short-term one that we'll get rid of 10 minutes after I've left the room. So we need to understand these things.
Yeah.
Of course, these will benefit society and people by understanding, if most therapeutic agents work at the molecular level, so if we can understand the instructions for origami, we can understand the instructions, but the way in which these are ignored by the body, then we can think about intervention methods. We can consider modifying genes and putting slightly more efficient versions of them into cases where we're unfortunate enough to have inherited a bad gene for a particular disease, or just been unlucky, because many of these processes of, for example, Parkinson's, Alzheimer's, they're either diseases of age where our protein network slightly falls apart, or they're just sporadic, they just happen just randomly. So if we can start to understand these, we can really form interventions so that the societal benefits are essentially something of a no-brainer, and that we're continuously hassled by different companies to, can you test this drug for us? Can you look at this? Can you think about this process, this molecular process, and provide us some instruction? But I actually genuinely think that the reason we should do this is just because it's just fascinating by itself. But, if we were thinking only about societal benefit, I would work in a drug company and churn out 1 million drugs and bind them and see which one cures A, B, C disease.
Right.
But understanding things at a fundamental nature is just so much more exciting, and also provides the models for the next generation of pharmaceutical approaches.
Right. Yeah, it takes out the guesswork.
It's amazing.
Exciting.
Thank you very much. Well, thank you very much, John. Thank you very much for your time. It's been great, and thank you very much for everybody for listening. Our website is very waffly, we've discovered, which is www.antiwafflepodcast.wordpress.com, and our Twitter account, which is @antiwafflepod. Please do get in touch because we'd love to hear from everybody. Thank you very much. Bye.
Bye.